The overall goal of this procedure is to screen small molecule libraries for agents that promote astroglial differentiation in glioblastoma stem cells.
This method can help answer key questions in the cancer stem cell field, such as what are the signaling pathways that are required for maintaining glioblastoma stem cells in the undifferentiated state. Proper execution of this technique allows for the straightforward identification of small molecules that promote glioblastoma stem cells differentiation. The implication of this technique extends toward therapy or diagnosis of glioblastoma, because reducing the percentage of glioblastoma stem cells may decrease the likelihood of recurrence.
This method can also be applied to studying differentiation into other cell lineages using other reporter constructs.
Our report is the first to establish a high-throughput approach to screen for agents that force glioma stem cells to differentiate.
Working with lentivirus particles in cancer cells derived from patients can be extremely hazardous. Therefore, standard procedure for working with blood-borne pathogens should always be followed.
To begin, seed 1 million glioblastoma stem cells, or GSCs, in 2 milliliters of complete neural stem cell growth medium in a 6-well plate. Next, transduce the cells with the lentivirus reporter at a multiplicity of infection equal to 5. To the cell and virus mixture, add 2 microliters of polybrine, such that the final concentration is 8 micrograms per milliliter, and incubate the cells at 37 degrees Celsius and 5% CO2 overnight. After incubation, replace the growth medium